Okay, good afternoon, viewers at home, and my hello. Can you hear me? Can you hear me from your device? What? Just join me. Can you hear me? Can you see my screen? What can you see on your screen? Okay, introduction to bioinformatics. Once again, good afternoon, uh, students and my viewers at home. This is another educational series of Omega Bio-Edutainment. So this is another opportunity to learn once again what bioinformatics is all about. Just like I told you earlier on, that bioinformatics is not just a course, it is a discipline. And whatever we call a discipline, it means that we need to take it serious. And that's the reason why up to today, it is one of the newest field where attention of the whole world is actually shifting towards. Um, just like I said, that this is not the first time we will be meeting in this course. It is a course that actually we have been doing all this while. So, and this is going to be a kind of a call to action. I am expecting you to have at least something to jot so that by the time I finish this presentation, you'll be able to have one or two reasons for this. So just like I said, bioinformatics is a discipline that combines interpretation, analysis, and utilization of biological data. And the earlier classes we had, I told you that bioinformatics, that is a biology aspect of biology that is dealing with informational molecules. And I gave you examples of informational molecules which are nucleic acid. That is, if you pick up a cell, when you pick up a cell, you have a macromolecule and you have micromolecules. Out of the micro and the macromolecules, we have protein, we have lipids, we have carbohydrates, and we have nucleic acid. Nucleic acid is in such a way that this is found inside the nucleus, and that's why they call it nucleic acid. And there are two types of nucleic acid. We have the DNA and we have the RNA. And you cannot, at this point in time, you cannot deal with anything bioinformatics if you cannot work with DNA, if you don't know the structure of DNA, if you do not appreciate the work of DNA. And the previous class, I think maybe last week, I told us in that presentation one of the reasons why bioinformatics is going to remain relevant till 2055 and beyond. And in that presentation, I told us that DNA, the structure of DNA, or the historical background of bioinformatics actually started from 1970. And those that label that actually plan the foundation of this could be credited to Watson and Crick when the double helix structure of DNA was discovered as far back as 1953. But it didn't gain any attention, no global attention, until the year 1977. That was when the first microorganism was sequenced, and that actually go on until 2003, when they sequenced the whole genome, the whole genes that are contained in human, and that is what they call human genome project. Now, as we go on, we are going to have a lot of benefits when we go on. Now, the next thing is what are the learning objectives of this course? That is, if we finish this presentation, if we finish bioinformatics at the end of the day, what are supposed to be the take home? What should you take back home, so that when you are lacking or lagging in any of these take home, you should know that maybe one way or the other, something is wrong somewhere. At the end of this training, you should be able to define bioinformatics. Not only that, you should be able to understand the relevance of bioinformatics in both microbiology as well as environmental biology. Another thing is that you should be able to utilize or explore key databases like NCBI, like GenBank, like Uniprot, like the KDDI, and so on and so forth. You should be able to make use of that to do bioinformatics analysis of your work. Okay, then apart from that, you are expected to also understand the role played by bioinformatics in solving some biological problem, and the attention of all of us is going to be shifted to when bioinformatics was actually used to solve the problem of COVID-19. We are going to see it. And deliberately, whenever you see any of these slides, and at the right corner, you always see an image. And that image can also be an additional source of information for you, because what we have said in the text is part of what the picture is also telling about. Okay, that is part of the learning objective is to be able to define, use your hand lens to magnify your knowledge, your definition of bioinformatics. After that, you'll be able to use relevance. How relevant is bioinformatics to you? Then apart from that, you should be able to use NCBI and GenBank to be able to analyze and retrieve your data. Then finally, you should be able to use bioinformatics to be a source of illumination for every one of us in solving biological problems. Now, the first thing is what is bioinformatics? That is, even if you don't know before, if somebody asks you, a layman is just telling you that you said you are doing bioinformatics, and what is that bioinformatics? What is the meaning of it? Bioinformatics is the use of computers to store, analyze, and interpret biological data. So, and that's the reason why we said that bioinformatics is more than just a course. It is a career. It is a career. It is a profession. Just like it is a profession to be a medical doctor, it is also a profession to also be a bioinformatician, because what they normally do is to be able to use computers to store, to analyze, and interpret biological data. In the earlier classes we had, I gave us I expanded that to mean 5 fingers, which must not lie. I told you that the first one is your ability to retrieve data. As a bioinformatician, your ability to retrieve data should not be what? It should not be joked with. Then, after retrieval of that data, your ability to manage that data is also very, very important. If you retrieve the data and you cannot manage it, that means you are not a good bioinformatician. Then the third one is your ability to be able to analyze that data. You analyze the data. So, and that is the third finger. The fourth fingers of your right hand should be your ability to interpret. You retrieve, you manage, you analyze, you do what? And you interpret. That is number 4, ability to interpret. Then the last one, which is your ability to utilize that data. If at the end of the day you retrieve the data, you manage the data, you analyze the data, and you also interpret the data, but that data you cannot utilize it, you cannot make use of it to make informed decision, that means you are not a good bioinformatician. So as we journey in this course, or in this discipline, or in this career, as we journey together, I want you to have these 5 key things at the back of your mind. That is, how can you retrieve data? How can you manage your data? How can you analyze that data? How can you interpret that data? And how will you be able to utilize the data? Then it is a field, it is a career in biology that combines biology, computer science, and statistics together. At least those 3 courses or 3 disciplines should be able to come together. When we talk of computer science, we are already talking about information technology. We are talking about information technology and the like. Then you should be able, bioinformatics is ability to decode DNA, to decode information about DNA, RNA, and protein sequences. Now, the question is, do we really need bioinformatics? You as a microbiologist, when you are coming in to study microbiology, none of you could have thought maybe a course like bioinformatics will actually come to be part of what you are going to do. So the question is, do you really need bioinformatics? You really need it because we already have biological data that are too enormous, that are massive, that are complex to analyze. So we need, it's just like an accountant that will tell you that it doesn't need a calculator. How possible is that? So the same way, if you are a biologist and you think that this course of bioinformatics is not relevant to you, that means you are not complete. And just like you see it in the image you can see this man is already worried. He has his face with his computer, and why is he worried? He's being worried because of the enormosity of the data, greatness of that data, diversity of that data, and there are lots of this, and that's why this man is still holding fast his part of his head. That is, he's really thinking fast on how to make use of a computer. So don't misjudge people, or don't misinterpret people that are biologists, that you know them, they are biologists, but they are moving around with their computers, they are moving around with their system. Don't say that this person actually loves to show off. No, it is the newest trend in the field of biology. Then we need this computer, we need bioinformatics, because whenever we use computer to analyze, to interpret our data, it is going to be more, it is going to be faster, it's going to speed up our level of comparison. So just like I told you that an accountant will never tell you that he doesn't need a calculator. No, because it's going to take that accountant a lot of time for him to be able to calculate all those figures, and that is the reason why, just like a calculator is not an accountant cannot live without a calculator, the same way that you as a microbiologist you cannot do without bioinformatics. Now, what are the application of bioinformatics and how can we get it? So both in the text as well as the images that you have, the image that you have here is attesting to the fact that we really need them. Number one, we apply our knowledge in bioinformatics for disease outbreak. I told you, like COVID-19. For example, now if there is any outbreak, what you do is to collect that person's sample. Whenever you collect the sample, you will do isolation and characterization. Whenever you finish doing that, the next thing you do DNA extraction. After extracting the DNA, the next thing is to amplify that DNA. When you amplify the DNA portion, that is going to give you a lot of DNA to have in your pool. And by the time you now do this pool, you now take it in form of a sequencing. You sequence this, and that's going to give you the totality of the alphabet of life, that is CTGC in DNA, in case of DNA. So that is why bioinformatics can be useful in the management of what in disease outbreak. Then in gene discovery, if you want to know, there are certain new genes that are being produced every time. For example, the gene that is making salmonella typhi to be more infectious than any other microorganism or any other bacteria is very unique to that gene, to that microorganism. And there are newer genes that are being produced every time as a result of changes in their environment. So if you want to discover all these genes, you really need the knowledge of bioinformatics. Another thing is that there are different types of drugs, different molecules of drugs that have been discovered, but there are still millions of them that are yet to be discovered. So it is with the help of bioinformatics now that new drugs are being produced. New drug candidates are now being detected. New drug candidates are now being managed. All around us, there are so many of the drug candidates that we live with every day. But because we do not have the necessary tool to be able to analyze this until we now have bioinformatics. So that is in the another area that bioinformatics is also very, very good. That is in drug development. Apart from that, it is also used by the environmental biologists. By the environmental biologists, what do they use it for? For environmental monitoring. So under normal circumstances, the environmental biologists, they normally use their knowledge in bioinformatics to predict what's going to be the weather, so that they will definitely know when to plant, when there's going to be infection of pests, when and how can they do that. So including planning for application of fertilizer and some other. So they normally use that. Then as also a microbial, as also a microbiologist, you are to use microbiology, I mean bioinformatics also, as a way of identifying new microorganisms. So that is why microbial identification is part of what you can use bioinformatics to do. Now, we just want to pick two real world examples. That is, even if you don't know any other thing, at least two real world examples that we are going to do should be able to explain why and how relevant is bioinformatics to us. The first one, the first real example is human genome project, human genome project, which contains full human DNA sequence. Now that as far back as the year 2003, all the alphabet that it can contain, that is made up of the genes in human, have been sequenced. It has been completed, and it is one of the application, real world application of bioinformatics that has actually given us that opportunity. Then the second one is the tracking of SARS-CoV-2. That is, now what they did in when there was a coronavirus, that is COVID-19. When that outbreak came in that 2019, it was as a result of bioinformatics. That was what helped us in, first of all, diagnose it. Apart from using our knowledge in bioinformatics for diagnosis, it was also bioinformatics informatic tools and experience that we use in the management and production of vaccine, and that is the reason why bioinformatics is still very very useful for us in the real world example. Then apart from that, there are, as a bioinformatician, at least as a foundational bioinformatician, you should get yourself to know and to learn, to be able to utilize. For example, now, if somebody is looking for any information, if you are looking for any information now, maybe about a project or about any topic, there are some people, the first place you go to is you go to the library. It could be e-library. Google is part of where you can search for it, because there are different information, different data that is already residing in that place. So as a bioinformatician, where can you fall, and what can you fall back to go and get your information? And that is the critical level at which, when you are looking for information about DNA, the first place that you should go to should be the NCBI, that is National Center for Biotechnology Information. So that is the first place you should go to. That is, if you are looking for any information that has to do with nucleotides, or if you are looking for any information at all, the first places where scientists will normally deposit data to is always NCBI. The next place where you can get your data is the gene bank. The same way they normally keep money in the bank is the same way that some data or biological data are residing permanently in the gene bank. So you need to also do that. And the last one, which is called the PubMed, that is whenever you are looking for scientific journals, scientific articles that has to do with that. So you need to you can go to that place and go and look for any information that is actually relating to that. Apart from that, now I want you to look at the activity that you can explore. How can you explore NCBI? Where can you get NCBI? How can you make use of NCBI? The first thing and the very quickest way to get it is that you could go to Google, type NCBI, it's going to take you there. But another thing is that in your browser, you can just enter www ncbi.nlm.nih.gov/ you is going to give you and take you there to NCBI. And when you get to NCBI, just type E. coli, or any microorganism that you are looking for their information. It's either you type E. coli or you type the organism you are looking for. And don't forget that the information you are looking for has to do with 16S ribosomal RNA. Don't forget that in the course I earlier taught you, I think first semester, I taught you microbial evolution and ecology. That was when I made mention of classification of that is modern classification of organisms. For that is a modern application classification of organism, which I said the domain of life, three domains of life. When I mentioned and I mentioned Woese, Carl Woese. Woese believed that the life can be divided broadly into three domains. The first one is a prokarya, the second one is a eukarya, and the third one is a archaea. Now, how was Woese? What method did Woese actually use? He made use of information about 16S ribosomal RNA. And also I told you one of the reasons why ribosomal RNA or RNA generally is more stable, is more useful than DNA sometimes. The reason being that RNA is more stable than DNA in terms of when DNA must have developed, or I mean, sorry, when DNA at a point in time, you just discover that it's very prone to mutation. Where DNA is mutated, you will see that RNA will still pass there without any problem. So that's the reason why. It's just like the 16S ribosomal RNA. I want to compare it. Let me just cite an example now. Please, what is your name? Elizabeth. What is your name? Lane. What is your name? What is your name? What is your name? What is your name? Are you related to? Are you related? You are not related. Now, but I mean, six of you, you are female. Abi? Now, let us now imagine that I don't want the full information about you. Let's say I don't want the full information about you. But one thing you know is that even if I see your finger, let's say I'm just picking one of your fingers, through that your fingers, I may be able to say, this one belongs to this one belongs to this one belongs to this, and this one belongs to that. So the same thing with what is called 16S ribosomal RNA. That 16S ribosomal RNA is like the fingers. You don't have access to the whole information. The whole information is going to be too large that you might not be able to actually work with, because the whole organism, like Microbacterium tuberculosis, it has more than 5 million bases. 5.5 million bases. Now, when you now have GCTT like that, you now have it in 5 million. So, for you to be able to manipulate that data or utilize that data, and don't forget that as a bioinformatician, I told you that you have to know how to retrieve the data. If it is too large, you won't be able to manage it. If it is too large, for example, now when you are dealing with a complete genome, sometimes if your internet is not that strong, if your internet is not strong at all, for you to be able to download a complete genome, honestly speaking, it might even take you 7 hours, 12 hours. So that is it. But if you just need a segment of that same organism, and that's what we call 16S ribosomal RNA. So that is just like you are getting parts, you are getting a finger of that organism, and your finger or your fingerprint is so unique. You know that there are no, including those um, twins that are identical twins, you don't have the same, they don't have the same fingerprint. So the same thing with 16S. So that 16S ribosomal RNA is like the fingerprint of microorganism or any organism at all. So if you have access to the ribosomal RNA, you already have it. So when you get to NCBI, you just type the organism you are looking for. If you are looking for Pseudomonas, type Pseudomonas originosa. If you are looking for Plasmodium, type it there. Whatever organism you are looking for, just type it there and type 16S ribosomal RNA. So the next thing is that you will just click on results, and by the time you click on that result, you just discover that you are already looking for what is called a metadata. Just like in Yoruba, meta means meta, that is three. And what are these meta? These are, you are looking for the organism, that is the organism name. That is where what you should be looking for. Then apart from that, you should be looking for the gene length, the length of the gene you are looking for. And the last thing you should be looking for should be accession number. That means any information that you are looking for, at least that three should be the basic that you'll be looking for it. So that is what we are saying. Please don't forget that as this lecture is going, I'm expecting you to make comments, like, and do the like, so that I will be able to know that truly you are following up with this presentation. Then after that, creative analogy, that is, just like I told you, that you should think bioinformatics to be in form of a library. And a library contains what? A library contains blue book. Okay, so bioinformatics is a biological Google where you can search for information, or what you can call the library, online library. So, and what you do is you will also get gene bank. Gene bank is like the library of the DNA. I've already told you information about that. Odelabi Nioluwa, I can see you. Okay, so can we go on? So now, in one sentence, at the end of this module, in one sentence, you should be able to define in your own words what bioinformatics is all about. What bioinformatics is all about. I told you that normally you should begin to think of bioinformatics to be in form of five fingers, which make up the very strong hand. Peace, Amos, I can see you. Okay, so the first one is that your ability to retrieve information. Ability to retrieve information. The second one is your ability to manage information. The third one is ability to analyze that information. Then the fourth one is ability to utilize the information. Okay, so that is that. So anybody that comes across you, to a layman, you should be able to tell them that bioinformatics is not just a course, it is a discipline, it is a career, it is a profession, it is something that is going to still be relevant in the next 30, 40, and 50 years, so to say. Okay, Akinla Mariam, I can see you. Adewumi Aderonke, I can see you. Oyediran Aminat, yes, can see you. Abdul Asis Mina, I can see you well. Anise Tomiwa, I can see you. Tavikat, I can see you. Adeotitimileyin, I can see you. Mustapha Basirat, yes, can see you. Olorun Fatima, I can see you. Now going back to what we have on the screen, we want to look at module 2, and module 2 is talking about biological sequences and databases. That is module 2. And what we have to do in module 2 is that, number 1, don't forget the central dogma theory. I have been telling you this. Central dogma theory. And the central dogma theory is a theory that believes that from DNA to RNA, then to protein. So that is very, very important. Apart from that, you should be able to also learn. Learn different databases. That is, where can you lay your hand into to be able to get information, to be able to get certain data. So you should be able to know that. So it is this second module that is going to expose us to know more about this. Okay, so now at the end of module 2, what are those things that are actually expected? What are those things that are expected of us? Number 1 is to identify types of biological data, that is, data that are DNA, RNA, and protein. You should be able to identify that. And those ones that deal with others like protein, you should be able to know them. Then you should be able to know that the format in bioinformatics, the general currency, let me use that word, the general currency of data, the general data currency in bioinformatics is faster. For example, so it's just like the way we have dollars for America, the way we have naira in Nigeria. So the same way, the same currency that we have for for bioinformatics. So your data should be retrieved. I told you that you know the ability to retrieve data, and it should be retrieved in form of what? In form of faster. So then after that, you should be able to now move between the gene bank and uniprots. I told you that whenever you are looking for nucleotide or any information, one of the databases is gene bank. And the next one, if you are looking for anything like protein, that is when you talk of uniprots. Okay, so you should be able to know that at the end of this module. That is, you should be familiar with sequence types, whether it is DNA, whether your sequence is DNA, whether your sequence is RNA, or whether your sequence is protein, that is in form of amino acid. Okay, or as a bioinformatician, it is easier. For example, which one is going to be easier to manage? Don't forget that one amino acid, 3 nucleotide sequences will be equal to 1 amino acid. So which one is going to be longer? Is it the sequence of DNA that is going to be longer, or the sequence of protein that is going to be longer? Which one? Which one? So I think you should know that the sequence of protein will be much shorter. It's going to be shorter than the. So it is. There are times that you retrieve your information in form of protein. Okay, and you should know this. I didn't say this. You should know this, that once you get any information, once you retrieve any data, the first thing is to clean up the data. Using that website to clean up the data, you clean it up, because there could be a lot of what is called junks on that data. That's one of the reasons. And whenever there is gaps, sometimes the sequencer, there could be gaps. And gaps in biology, it has a different meaning. Gaps means mutation by deletion. And I'm very sure that in your first year and your first semester here, I taught you fully on what mutation are, and the different types of mutation, and the causes of mutation, and mechanism of mutation. But that is not the focus of our discussion today. Then you should be able to learn how to retrieve data. Your ability to retrieve that data is very, very important. You see, For example, now if I should leave you all now and I ask you to go to Google, go and get any information, the way each of you will retrieve that information will be quite different from another person. And that's why retrieval of information is as good as managing the information, analyzing the information, utilizing the information. Because once they said a journey of 1,000 kilometers will start with just a step in the right direction. So if you do not know how to retrieve data, you will get lost at the end of the day, and that is not going to be good. That is why under normal circumstances you should be able to know how to retrieve data. It is part of what we are going to learn in this module 2. And just like I said, by now you should know the difference between DNA, RNA and protein. You should know that DNA contains ATCG, that is adenine, thymine, cytosine and guanine. And you should know that it is RNA that contains adenine, uracil, cytosine and guanine. And whenever you talk about protein, you should know that protein contains 20 amino acids, and it is this amino acid that we combine together to form the structure of a protein. Okay. Now, looking at what we have on the screen, we have a kind of a test format, the FASTA format. How can you know, how will you identify that the currency, for example, now, if you want to identify the currency of any country, just like I told you, that FASTA is just like a currency in bioinformatics. So the only way you can recognize a currency of any country is that you'll be looking for logo of that country. So the same thing, the logo of FASTA is greater than sign. Whenever you see greater than sign, it is FASTA format that is going to start with greater than sign. Okay, and there will be, so that is what you have as you have it on the screen. So that is, so even if you have only the nucleotide sequences, so if you want to convert it to data format, I mean FASTA format, the only thing is just on top of it, just add greater than and write the name of what you are looking for. Okay, that means you have converted it to raw data into FASTA format. Then the sequences, major sequences, different types that we have. We have gene bank, which I said is where you can find information about DNA and RNA, and that one is that one was established. I mean, sorry, the right owner of gene bank is the whole world. The whole world is just depositing data there, and that data has now become so enormous that whenever anybody from any part of the world is looking for any information, you go to the gene bank. But there are certain countries, for example, those of them in the Europe now, they are now having a different database in the sense that even if they submit the information, that data into the gene bank, they will still have a copy in their own region. It's just like somebody, when you imagine Nigeria as a country, as a federation country that is divided into federal government, state government and local government. So under normal circumstances, if you need any information, it's just like you cannot go directly to federal government. Even if you go to federal government, you still trace you back to the local government. So information about you can be traceable in your local government, and that information can also be found in the national, because the information in local government will only be specific for that local government. For example, if you are talking about Akinyele local government now, that means the information that will be available there will only be based on that Akinyele. And the same way, We have information deposited at the gene bank where everybody around the world, every continent is depositing their this thing there, their information there. So we have some Europe, we have some from Japan, and we have some like a protein. So DDBJ, that is the data bank for those that are from Japan. Europe, they have their own. Protein, those one they also have their book. And anatomy of gene bank record. When you go to gene bank, what should you be looking for? I told you the first thing is that you are going to see an accession number, the specific number that will be assigned to that organism. So it's going to be there. Then the source organism. What is the owner? Which organism is the origin of the databases or the sequence you are talking about? That will be seen. For example, the one that is being displayed, you can see that the accession number is right there, which is MN826190. That is the accession number. Then the source organism is Escherichia coli, and the sequence features like the source. And if you look at this one, the length of this is going to be 1389, okay, sequences. Then the references, that is the first person that deposited that sequence there, the name will be there, and that is going to be the author. And as you have it displayed on the screen, the author of this one is just Mocking K. Jenkinson, as well as other things. So now it is for you to be able to navigate and make use of the gene bank. So just like I told you, then faster format I told you is just like the currency of bioinformatics or biological data is faster. And if you want to test for the originality of any product at all, it is for you to be able to test it using scanner. When you scan, it's going to tell you if you want to know any drug, whether any drug is original or fake. That is when you use there are some that they have unique number that by the time you scan them, it's going to tell you whether it is original or it is not. So whenever you see faster, whenever you see any faster, the component of any faster is going to be outside. It's going to have so we are going to see this. Then again, just like I told you that by the time you will be working in pairs, that is two, you'll be working in pairs, you should be able to be cognizant of what and what should be expected for you. That you work in group, then you need to take note of the gene, the name of the gene, you need to record the length, you need to tell us about the organism, and you need to tell us more about the features of that organism right away from NCBI. Then apart from that, the last module we want to talk about is module 3, and that is going to be where we are going to stop today. I told you that normally, the normal thing is from this course supposed to be module 1 to 13. Module 1 to 13. And previously, in this course, I want to believe that we have actually done module 1 to 2 before, and today was supposed to be just for module 3, and that is what we want to do. I just want to explain. You will do it during the practical session now. You will do it. So module 3, we'll be talking about what is called sequence alignment. Sequence alignment. Sequence alignment. When you are talking about alignment is when you are combining and comparing. That is, you are aligning there. For example, you can have one sequence. You have one sequence. Let's say, I hope all of you are from microbiology. Okay, let's say now in this your phone, you want to know the microorganisms that are present in your phone. What you should do is to go and get a very clean swab, sterile swab. Then you use the swab to collect, just to rub the surface of your phone. And by the time you rub the surface, You just take it to the laboratory, you prepare agar, you use different types of agar to isolate the organisms that are there. Those microorganisms that are there, you need to start making use of maybe biochemical tests or other tests, laboratory tests, to be able to know. So you be able to know whether they belong to E. coli or this or that or that. So by the time you now do that, that is wet lab. By the time you do that, the next thing, once you are able to get the pure clone, is to do DNA extraction of that. When you now do the DNA extraction, the next thing you will do is to sequence it. After DNA extraction, you apply PCR, then you sequence it. And don't forget that the sequence that is very, very common is that of 16S. We call it partial sequencing. 16S ribosomal RNA. So by the time you now do the sequencing, partial sequencing, you have some set of sequences. So now you now have these sequences, but you don't know, you want to compare, you want to know whether your sequence is similar to what is available in the gene bank. You know, I told you that information about organisms around the world are being deposited there. So by the time you now get one sequence and you are comparing it with a sequence anywhere in the world, that means you are comparing it with a alignment. And anywhere we are looking for similarities. We are looking for homology. And what do we mean by homology? Homo, same, logy, origin. Okay, that means we are looking for whether your sequence has homologous relationship with other sequences around the world. So when you now check the level of alignment, if it is greater than 99%, that means that organisms, they are almost the same, if they are not 100% the same. But once you have below 70%, that means that organism is not, they are not related. Evolutionarily, they are not related. So that is what we call sequence alignment. That is, we want to, when you have more than one sequence now, you want to bring them together. You want to bring them together and see where they are, where they differ and where they are not. So at the end of this, you should be able to understand what we mean by sequence alignment. You should be able to differentiate between local and global alignments. You should be able to have a practice of a manual alignment, that is the one you can use with your pen and pencil. I mean paper and pencil. You can use that. Then you should be able to know if you want to perform alignment that involve more than three, four, five different sequences together. So for you to be able to have one, I will start ticking it. This one, no, this one, this one. It's going to take a long time. So how can you use computers to be able to, what is that tool that you can use? We are going to see it. It is called emboss needle. We will see it there. So I have explained to you that a sequence alignment means comparing two or more sequences to find their similarities. That is a sequence alignment. We are comparing two or more sequences together to find similarities. Then another one is that you are trying to help to identify if there is any evolutionary relationship. To identify if there is any evolutionary relationship, then you want to use it in gene and protein. That is, you can use alignment to detect the function of that kind of a protein. And there are basically two types of alignment. The first one is called global. The second one is called local. The global means there is a full length alignment. Okay, when the alignment cover a very long sequences, that is called global. And if it affects only partial alignment, if it is aligning partially, it is called local. And don't forget what I told you about blast in the last lecture. I told you that blast means basic local alignment sequence tools. BLAST, basic local alignment sequence tools. So by now you should be able to know how to blast any sequence that you have. So apart from that, hands-on example, you have two sequences that is already given to you, and you have to bring them together and find how similar they are and wherever they are not similar. And the tool, computer tools that you can use for alignment is called emboss needle. Emboss needle, you will see that. Then you just enter the sequences, sequence one, sequence two, sequence three, until that you just click submit. It is going to do it for you. Or if you don't want to use emboss, you can use cluster omega. It is right there. It's still going to perform the same function. Then the most advanced tool that you can use is called MAFT. So that is beyond the scope of this work. So trying to do this, you have to perform an activity that will involve emboss. That is, you just type in emboss, www.ebi.ac, and so on and so forth. Then you paste your sequences there. By the time you paste your sequence, you click on run. By the time you now run, you will now start observing whether there is matches, whether there is any matches or there is no. Then gap, you will be able to also discover that. Then I told you, whenever there is any match, if there is any match, it will be recorded as 1. If there is gap, you record it as minus. If the identity, the percentage is high, 99%, that means it is exact. And the number of matches you have all over the total length is going to give you how you are going to do it. And this one is also giving us how you can imagine alignment from two sentences. Okay, I love bioinformatics, and the second sentence is I love biochemistry. There are certain things that are common. I is common in the two, love is common in the two,